Welcome to General Biology. Today's video is going to be on mitosis. So one easy way to remember the stages of mitosis is we call it a pee-pee mat. So I always think of like a big mat that a dog, if you lived in a big apartment complex in New York City and you had a dog and you didn't want to necessarily get up super early to walk them, they have mats that you can train your dog to actually go to the bathroom on, and then you just dump the tray. So it's a little easier if you live on the 40th floor or whatever. What we're looking at with mitosis is basically once we come out of interphase is not really part of mitosis, but once we come out of interphase, then you're looking at prophase, prometaphase, and then metaphase, anaphase, and telophase. Prometaphase is something that they've started talking about in all the new textbooks, and so I added it to this particular presentation. So prior to us going into this process, one of the things we're going to need to do is at the very beginning of prophase is we need to condense the DNA so it's easier to separate, so it doesn't become a tangled up mess. And so what happens is the DNA will wrap around histones, and then it will coil, and then that coil will coil, and then that coil will coil again into a chromosome. And so they call it coiling and supercoiling, and I call the last one super duper coiling. So when we get into prophase, all of the chromosomes have been condensed, okay? And we're going to move our centrioles to the opposite ends of the cell. Basically, the spindle fibers grow, and as they grow, they're pushing the centrioles apart. And then the centrioles will anchor to the cell membrane on the opposite ends of the cell. And at the very end of prophase, the nuclear membrane is going to break down so that the spindle fibers can grab onto the chromosomes. This is what chromosomes look like at the end of prophase. They're kind of spread out throughout the cell. At that point, spindle fibers will grab onto the chromosomes, and we call this prometaphase, when the spindle fibers grab onto the chromosomes and move them to the central plate or the center of the cell so they're getting ready to be separated. Once they officially reach the middle of the cell, we call that metaphase. So they're going to line up in the middle, and that way when all the sister chromatids separate, they can be pulled kind of cleanly to the opposite ends of the cell, and we end up with one of the sister chromatids from each chromosome at the opposite ends of the cell. When the separation starts, we call that anaphase. So an enzyme called separase will go through and divide all the centromeres, and then we will be pulled to the opposite ends of the cell. And that process of pulling them to the opposite ends of the cell, we call that anaphase. Ana for apart, and it's the stage from when they first separate until they get to the far ends near the centrioles. Once they get to the ends, then we roll into telophase. In telophase, the new nucleus will start to form around the chromosomes as quickly as possible to protect that DNA. And then you will start getting the spindle fibers will break down. Once the new nuclei have formed, then the chromosomes will break down back into their linear form, the chromatin we call that. And then the cell membrane will start to cleave, and if it's an animal cell, it will pinch off, and we call that cytokinesis. And if it's a plant cell, it'll form a cell plate, which I'll show you in the next couple of slides. So with an animal cell, what happens is a fiber of actin will start shortening, and it will cause cleavage in the cell, and it will get shorter and shorter and shorter, kind of like twisting a big long balloon. if you start twisting it, they get skinnier and skinnier and skinnier in the middle, and then pretty soon it looks like two balloons. So that's what's going on with animal cells. And then in plant cells, what will happen is you will start depositing vesicles of cellulose along the center of the two cells, and then those vesicles will start growing together and forming a cell plate. And then eventually that cell plate, those vesicles will get thick enough that they actually bind to the cell walls on either end, and we get a new cell wall down the middle of the cell. Here's a picture of that process happening. You can see a bunch of vesicles being deposited of cellulose along the middle. Eventually all those vesicles will combine with one another and then combine with the cell wall on the margins, and you'll have a new cell wall going down the middle of your two plant cells.